Deoxyribonucleic acid, or DNA, is a long, double-stranded molecule that contains the genetic information for all living things. It is made up of 4 nucleotide bases: adenine, thymine, guanine, and cytosine. These bases pair up in a specific way. A always pairs with T, and C always pairs with G. The sequence of these base pairs determines the genetic code that contains the instructions for building and maintaining an organism. The discovery of DNA is credited to a number of scientists, including Gregor Mendel, who conducted experiments on pea plants to determine how traits are passed down from generation to generation. Before we continue, please click the like button, subscribe to the channel, and activate the notification bell. However, the most famous scientists associated with the discovery of DNA are James Watson and Francis Crick, who in 1953 proposed the double helix structure of DNA. Watson and Crick's discovery of the double helix structure of DNA revolutionized the field of genetics and led to new understandings of how traits are inherited. Their research was based on the work of other scientists, including Rosalind Franklin, who used X-ray crystallography to study the structure of DNA. DNA is organized into structures called chromosomes, which are located in the nucleus of a cell. Humans have 23 pairs of chromosomes, for a total of 46 chromosomes in each cell. Other organisms, such as dogs and cats, have different numbers of chromosomes. The function of DNA is to store and transmit genetic information. When a cell divides, the DNA must be replicated so that each new cell receives a copy of the genetic code. This process is essential for the growth and development of an organism, as well as for the repair of damaged cells. In addition to its role in inheritance and cell division, DNA also plays a critical role in protein synthesis. DNA contains the instructions for making proteins, which are responsible for carrying out many of the functions in the cell. During protein synthesis, the DNA is transcribed into a molecule called messenger RNA, mRNA, which is then translated into a protein. DNA can also be used in a number of ways, including forensic investigations, gene therapy, and the diagnosis and treatment of genetic disorders. For example, genetic testing can identify mutations that cause diseases like cystic fibrosis, Huntington's disease, and sickle cell anemia. Researchers are also exploring the potential of gene therapy to treat these and other genetic disorders. However, an alteration in DNA can result in genetic disorders, which can have serious consequences. These disorders can be caused by mutations that occur spontaneously or are inherited from parents. Some examples of genetic disorders include Down syndrome, cystic fibrosis, and sickle cell anemia. In conclusion, DNA is a complex molecule that contains the genetic information for all living things. Its discovery was a major breakthrough in the field of genetics and has led to new understandings of how traits are inherited. Understanding DNA is essential for advancing our knowledge of genetics and developing new treatments for genetic diseases. However, genetic disorders can have serious consequences, and it is important to continue researching and developing new therapies to treat these disorders.